Hello, everyone. Good morning for those here in Brazil. Good afternoon for those overseas. This is the second technical session here in Brazilian Symposium on Bioinformatics. We have for this session 3 papers, and I would like to make you know that the presentations is going to be in a sequence, and then at the end, the presenters can answer the questions. So for that, I ask you that if you have any question, please write it down in the comments on YouTube, and then I can read them for them. And well, let's start with the first presentation. I would like to invite the first presenter to share the screen.
Can you see it?
Yes, we can see it.
Perfect. Okay, hello everyone, and thank you for giving me the opportunity to present this work here. So as the title says, I'm going to talk about orthology and xenology, which are 2 different types of relationships of genes. And the main idea is that you can estimate them separately from gene sequence data. And the question that we will be concerned with is when are they compatible in the sense that they can be explained by a common scenario. So this is joint work with these people, Mark Helmuth, Mira Michel, Nikolai Neugard, and Peter Stadler. So that we are all on the same page, I first want to give you some background on our model, how we imagine evolution to progress. So this is basically a summary what Mark already told you in the last session. So we assume that there is some underlying true scenario with a species tree. So we do not consider networks. We have a set of extant species, which are all descendants from a common ancestor. The evolution of the genes as part of the genomes of these species is closely tied to that of the species. So a gene tree is embedded into the species tree. And whenever we have a speciation, then this also leads to a divergence in the gene tree. But you can also have events that are not associated with speciations, like duplications, gene losses, and horizontal gene transfer. And according to these types of events, we classify pairs of genes. We say that they are orthologous if their last common ancestor was a speciation, as for example, A1 and B1 here. We say that they are paralogs if it was a duplication, like A1 and A2. And finally, we have xenology, which is defined a little bit differently. We say that 2 genes are xenologs if on the unique path that connects them, there is some transfer, as for example, for A3 and D here. And here we will actually be concerned with a refined version of xenology, where we also care about the site from the last common ancestor where such a transfer happened. And knowing these so-called homology relations is of interest for several reasons, and we've also heard this already in Mark's talk. For example, orthologs are interesting for genome annotation and phylogenomics. And there's only one big problem, and that is that we are not able to directly observe these homology relations, nor the complete history of a gene family. So the main task here is to reconstruct the relations based on the observable genes, which are the ones that are in the extant species. So there are several methods how this can be done. You could, for example, try to explicitly reconstruct such a scenario, but there are several issues with that. For example, reconstructing gene trees is still a quite difficult issue, and also finding such an embedding, so the reconciliation, is computationally expensive, and therefore we will here focus on methods that avoid reconstructing trees. So the Mark called them tree-free magic. And usually to do this and to verify that what you've estimated is valid, it's usually crucial to understand the mathematical structure of these relations. As for example, we've also seen for the interplay of best matches and orthology. So here we will be concerned with two relations, and that is orthology and xenology. And for both of them, there exist tree-free methods to estimate them from gene sequences. So for orthology, this would be the graph-based inference method. On the side of xenology, we have, for example, the group of implicit phylogenetic methods. Both of them are not perfect, but for the moment, let's assume that we can use them to get estimations of both relations. So in the first part of my talk, I will talk a little bit about the structure of orthology and xenology and generalizations of them. And in the second part, I will turn to this question, when are they compatible, and answer this from a mathematical point of view. So let's get started with orthology. Orthology is defined in terms of event-labeled or vertex-labeled gene trees. So we assign to all inner vertices a specific type of event, like speciations. And we say that two leaves are orthodox if their last common ancestor was a speciation, as for example, A and C. So this is defined in terms of last common ancestors, and therefore symmetric relation, and can be represented by an undirected graph, which I've drawn here for this example. So we have an edge whenever two genes are orthodox. Okay, when you look at this, you might say, okay, we lose a lot of information when we consider this orthology graph. Because for two genes, we only know whether their last common ancestor was a speciation or not. But you may also want to know the types of events for all other pairs. So there's still some room for generalization, and this brings us to so-called symbolic ultrametrics. And they are also defined in terms of vertex-labeled gene trees, where we assign to every inner vertex some event from a set M, which contains speciations and so on. And instead of relations, we now consider maps delta that assign to pairs of genes one of these types event. And we simply do this by assigning to all pairs of leaves the type of event of their last common ancestor. So this is still a symmetric map, and it can be represented by an undirected graph, except now we have a complete graph, and we color all edges according to the type of event at their last common ancestor. And a map that can be explained by a tree in this sense is called a symbolic ultrametric. And these maps have been characterized more than 20 years ago by Becker and Dress, and they found three sufficient conditions. So the first one is the symmetry condition, and the second one is the co-graph property, which says that if you consider one color at a time, then you cannot have induced paths on four vertices. So you can't have something like this. And the reason for this is that if you consider only one type of event, then you're essentially back with orthology, and for orthology, P4s were the forbidden induced subgraph. And finally, you cannot have something like this, three vertices with three differently colored edges. So by definition, every symbolic ultrametric is explained by a vertex-labeled tree. But in general, there are many trees. And if you look at this example here, then this is in a specific way not minimal, because here we have an edge whose endpoints have the same label. So what we could do is we could simply contract this edge into a single vertex like this, and give the resulting vertex the label of the original endpoints. And it's quite easy to verify that this changes nothing about the symbolic ultrametric that it explains. So it turns out that we can continue this for such edges. Here we are already done because there are none left. And if we do this, we will eventually end up with the unique least resolved tree of the symbolic ultrametric, which was also shown by Becker and Dress. So this is the first thing that I want to highlight. if we have some map delta of this form, then we can check in polynomial time actually whether it's a symbolic ultrametric, and also construct its unique least resolved tree T delta, in which case this vertex labeling T delta is also unique. So much about symbolic ultrametrics. Let's now turn to xenology. This is also defined in terms of labeled gene trees, except now we have an edge labeling, and we label every edge either as a transfer edge or as a non-transfer edge. So we end up with edge label trees that look like this. So this is simply an illustration how a corresponding scenario could look like. And we say that a pair of genes X, Y, an ordered pair, is a xenology pair if the path from their last common ancestor to Y, so to the second one, contains a transfer edge. So this is no longer symmetric, because if you swap these, then you look at two different paths. So we have to represent it by a directed graph. So let's look at some example. For example, the ordered pair AC, then we have to look for their last common ancestor, which is here, and consider the path to C. So there's no transfer here, and therefore this edge from A to C is missing. In contrast, if we look at the pair CA, then we have again to look for their last common ancestor and the path to A, and there is a transfer here now. So the edge from C to A is present. If you look at this, you might again say this is not really general enough, because you also want to model other types of events that are associated with edges in the gene tree. So let's also generalize xenology a bit, which leads to so-called Fitch maps. These are also defined in terms of edge-labeled gene trees. And now we assign to every edge a subset of events. So N is now a different set of events with transfers and so on. And instead of relations, we now again consider maps that assign to ordered pairs of genes also subsets of these events. And we do this in the following way. If we want to know epsilon of AC, and we have to look for their last common ancestor, which is again the root, and then collect all the types of events on the path to C. So in this case, that would be the red one, the blue one, and the green one. In contrast, epsilon of CA, we look at the other path to A. Here we see the blue one, the red one, and the yellow one. So we can continue this for all pairs, and then we end up with the Fitch map that is explained by this edge label tree. So Fitch maps have been characterized by Helmut Seemann and Stadler, and I don't want to go into the details of the characterization. I only want to point out this result here, that this says that, like symbolic ultrametrics, Fitch maps are also explained by unique least resolved trees, T epsilon, lambda epsilon. And here least resolved means that you cannot contract any edges, but you also cannot remove any of the assigned events. So more formally, the sum here, which is loosely speaking the number of stars, is minimal. And Fitch maps can also be recognized in polynomial time, and we can also construct this tree T epsilon. So this is the second thing I want to highlight. For Fitch map, we can get its unique least resolved tree T epsilon. So with all of these preliminaries, I now come to the main results of the paper. And this is centered around this definition here, which formalizes compatibility of two such maps. So here we consider pairs of maps delta and epsilon that assign to pairs of genes, either single events or subsets of events. So this delta is associated with the vertex labeling in the tree, and epsilon is associated with an edge labeling. And we say that a pair of such maps is tree-like if there is a tree T with vertex labeling lowercase t and edge labeling lambda, such that this here explains delta and T lambda explains epsilon. So from this definition, we already get two necessary conditions for tree-like pairs of maps, because this here says that delta must be a symbolic ultrametric, and this says that epsilon must be a Fitch map, simply because they're defined in this way. Now it turns out that these conditions are not sufficient. So to derive some more and to answer this question when a pair of maps is tree-like, let's summarize what we already know. So we know that delta must be a symbolic ultrametric. Therefore, we can get its least resolved tree T delta. And on the other side, epsilon must be a Fitch map. So we can get another least resolved tree T epsilon. And since both of these least resolved trees are unique, this means that a common tree that explains both maps must be a refinement of both of these trees. So from this, we can now get a third condition that looks as follows. Both of these trees must admit some common refinement because otherwise this is simply not possible. So we could show that these conditions are actually sufficient now. So we get the following characterization of tree-like pairs of maps. A pair delta epsilon is tree-like if and only if delta is a symbolic ultrametric, epsilon is a Fitch map, in which case these two trees exist, and also they must admit a common refinement. So if we look, let's look at some example. Here we have a tree T delta obtained from a symbolic ultrametric, and then another tree T epsilon. Do they admit a common refinement? Okay, we could simply here take C and D and refine this tree by collecting both of them under a newly created vertex. Or equivalently, we could go in this tree and collect A and B under a newly created vertex. So in both versions, we end up with this tree here. So this is a common refinement of these two trees. And it turns out that this is actually the minimal common refinement of these two trees. And it turns out that for the minimal common refinement, we always find a vertex labeling T star and an edge labeling lambda star, such that this triple here explains both maps. So this means that there is always, for tree-like pairs of maps, a unique least resolved tree. So this is quite nice, and the characterization also tells us that tree-like pairs of maps can be recognized in polynomial time, because the first two steps can be checked in polynomial time, and constructing a common refinement, if it exists, can actually be done in linear time. So the total complexity is actually dominated by the second step, and so since this is the set of events, if this is bounded, then we end up with quadratic complexity. So if you look at this, when you've constructed this, and if you look at it, you might want to again take a step back and ask yourself, does it actually make sense? And by this I mean, for example, if we consider the red stars as transfer edges, does it make sense that this transfer edge here emerges from a speciation event? And I think most would agree that this doesn't really make sense. So we might want to constrain the label trees that we're looking at or that we're interested in a little bit further. So this is what I want to talk about in the remaining time. So for simplicity, let's go back to transfer edges and non-transfer edges. So one constraint that we might want to introduce is the one that I mentioned on the last slide. If we have a transfer edge UV, then this should only be possible if the upper vertex U was an HET event. And then a second constraint that is commonly made in the field is motivated by reconciliations, because for reconciliations, so an embedding of the gene tree into the species tree, and for these you would usually want to find someone that requires a minimal number of events that explain the reconciliation or that are necessary to explain it. And if you look at this local reconciliation here, then both edges leading to the children of vertex U are transfer edges. So it doesn't really make sense to place U in this part of the species branch, in this branch of the species tree, because we could as well place it here and then either directly come from above or make the edge to its parent a single transfer. So in any case, we can save at least one transfer. So how can we translate it into our edge-labeled trees? Okay, we could introduce a constraint that looks like this, which says that every inner vertex has at least one child such that the edge to this child is a non-transfer edge. So whenever we have transfers, then we also want at least one child that is mapped in the species tree below U. Okay, what does it now mean for our tree-like pairs of maps? For a pair of maps delta epsilon, of the form that we've already seen, then we would want an explanation for this pair of maps that also satisfies constraints C1 and C2. So at the first glance, this looks like a more complicated problem than before, but it turns out that there is some explanation that satisfies C1 and C2, if and only if also the unique least resolved tree satisfies C1 and C2. So since this can be constructed in polynomial time, we can also easily check whether it satisfies C1 and C2. And if it doesn't, then we can stop searching because we know from this result here that there is no other explanation that also satisfies C1 and C2. So with this, I'm already at the end, almost at the end of my talk, and I want to close with some open problems. Here we have only considered the directed Fitch maps, but there is also a symmetrized version where we do not care about the site from the last common ancestor where some event happened, but only that it happens somewhere on the path between two genes. And even recognition of such so-called symmetrized Fitch maps is an NP-complete problem, and therefore checking whether they are compatible with other information, such as trees or orthology, is probably not much easier. And the second point here is reconciliations. We haven't considered them explicitly here and only used them to motivate certain things like the constraints. But if you do consider them explicitly, then it's known that they impose additional constraints. And these are also not simple ones that are can be checked locally, but have a more complicated structure. And then the final point is, of course, the inference, where there is a lot of room for improvement. And actually, inferring the dialectic xenology problem is to a large extent an open problem. But before you have too many question marks, why it already makes sense to look at compatibility of such relations, one motivation that we have is also that it seems promising to get dialectic xenology relations to include other available information, such as orthology or best matches, which are associated with some information on the tree. So you see that there's so in this way you could imagine to make the xenology compatible in the construction process, or at least make statements on the ensemble of compatible relations that also agree with the data. So you see there's still lots of work to do, both for people who develop and implement the methods and for more theoretical people. So with this, I want to thank my co-authors, and of course also thank you for your attention, and I'm happy to answer any questions later.
Thank you very much, David, for your presentation. So if you have any questions, please write down on the chat, and we're gonna take them at the end. I would like to invite the next presenter to share the screen and start the presentation, please.
Other? I'm ready to start. Okay. So my name is Rodrigo Henrique Ramos. I am a PhD student in computer science at the University of São Paulo, and I would like to thank the organizers of the BSB event for the opportunity to be here, talking about our work, topological characterization of cancer driver genes using reactome superpathways networks. Let's start by breaking the title in three parts, so we can understand what is a topological characterization, what are cancer driver genes, and what are reactome superpathways networks. Starting with cancer driver genes. Cancer is a molecular disease caused by genetic mutation that leads to cell uncontrolled growth and division. Tumors undergo a large number of mutations, but only a small portion of them contributes to the oncogenesis and tumor progression. This concept leads us to the idea of driver mutation, that are this small portion of mutation that leads to cancer, and passenger mutation, that are the majority of mutations that does not lead to cancer. An open challenge in cancer genomics is the identification of cancer driver genes, that the genes associated with cancer. We have some databases that make available lists of driver genes. One of them is the InfoGen. Here we see the cloud with the name of some genes, one famous cancer driver genes. And we also have the NCG that has a list of genes. There is a high intersection between these two databases, and we use the union between these two. Hectome Superpathways Networks. Hectome is a pathway database, and pathways are a set of genes that interact to perform specific biological functions. Hectome has more than 1,800 pathways, and in a recent paper, they group all these pathways into 26 superpathways, and these superpathways represents major biological functions. For example, cell cycle, circadian clock, programmed cell death, and reproduction. Here, we see the 81 genes Hectome associates to the reproduction pathway. But since we have the information from the other database about cancer driver genes, we can identify that in these 81 genes, 14 of them are associated with cancer. And since these genes interact with each other to perform biological function, we can model them as a network. So here we have a network with the same 81 genes we had before. But now a gene is not just a member of a set. He's a node in a network, and this node has topological features like the number of neighbors, its position in the network, a cluster, and something like that. And once again, we can identify in this network which genes are associated with cancer. This is the same plot as before, just to stretch a little. And here we can see that we divide the network in two groups. The red nodes are the cancer driver genes, and the blue nodes are no drivers. We don't know if maybe in the future one of these blue nodes will be discovered as a cancer driver gene, but we don't know the information yet, so we have driver genes and no driver genes. To further illustrate the concept, we show another network that is the programmed cell death superpath, and once again we have the set of drivers and the set of no drivers in the network. Now that we can model biological functions as networks and identify in these networks which genes are associated with cancer, we need a way to analyze the nodes. And I will present here four centrality measures as approach to topological characterize the nodes. I use a sample network. It's not a pathway network, okay? It's just an example. And the first centrality I'm going to use is the degree centrality that measure the number of neighbors a node has. So if we look at this network, each node has a value, a score, meaning the number of the neighbors. And this node and this one, they have the same score. They have 8 neighbors. So they are equal if we look at the network at a degree perspective. But when we change the centrality measure, we change how we look at the network. And using another centrality measure, the closing centrality, that measure how close a node is to every other node. So from this node, the shortest path to any other node. We see that this node is more close to the other nodes in the network than this. So they have the same degree, but when we look at the closeness centrality, they change score. Another centrality you use in this work is the clustering centrality that measure how the neighbors of a node are interconnected. The node that has 8 neighbors has 0 cluster in this case because none of their neighbors are interconnected. This node also has 8 neighbors, but some of their neighbors are interconnected, but not all. So the cluster is between 0 and 1. Lastly, we use the betweenness centrality that measure flow of information. If you look at all possible shortest paths between all nodes, many of these paths go through this node, so the betweenness is high. Betweenness, once again, measure flow information or bottlenecks, if you want. It's an interesting measure. So with these three things in mind, topological characterization, cancer driver genes, and superpathways, the objective of this work is to investigate the hypothesis that driver genes are topologically different from other genes in the same pathway. To achieve the objective, we join data from three different databases. We already talked about the drivers database and the Reactome pathway, but we also use the Reactome functional interaction network. It's not a pathway network, the network that tries to model the interactome of a cell. And it has 14,000 nodes. It's a huge network compared to the ones we use here. And from this network, we extract induced subgraphs using the genes set and enrich these networks with driver information. So in the end of this pipeline, we end up with 26 super pathway networks enriched with cancer driver genes. We analyze the 7 most pathways associated with cancer. How we did that? For example, programmed cell death, Hectome associates to this pathway 216 nodes. But when we extract from the network I induced subgraph, this network ends up with 201 nodes on its large connect component. From these 201, 27 of them are the drivers, which represents 13%. So we did this to the to all the super pathways and select the 7 most associated. And we also did a consensus network. We call all super pathways. We get the nodes from all the 26 super pathways and create a network with 11,000 nodes on it and 649 cancer driver genes. The first analysis we did was to compare the centrality distribution among the drivers nodes and no drivers nodes. So, for example, here at the programmed cell death pathway, super pathway, looking at the degree distribution, we see that the blue box represents the degree distribution among no drivers, and the red box represents the degree distribution among drivers. We see there's a difference in the box, a difference in the median. But since the drivers are a subset of the entire network, what we see here can be explained by random sampling. So we did these orange boxes that represents one sampling, 10 sampling, and 100 sampling. So when we look at the blue and red box, there is difference. But when we compare the median of the red box with the median of the orange boxes, the median can be explained by the random sampling. Not the entire distribution, but the median. When we look at the reproduction pathway and degree, we see a difference. The distribution of drivers is different from no drivers and from random sampling. Returning to programmed cell death, we see that the clustering is lower, the betweenness is way higher than the no drivers and random sampling, and the closeness is also higher than no drivers and random sampling. Excluding DNA repair, that we did not see a significant difference between drivers, no drivers, and random sampling, all others pathways show some kind of differentiation among the centrality, as we can overall say that drivers are hubs because they have a higher degree, and they connect different parts in the network because they have a lower clustering, and they concentrate flow of information because they have, in all cases, a higher betweenness, and they are more in the middle, the center of the network, because of the closeness. We also show here the, the comparison between the others networks. And we also analyze the differentiation between drivers and no drivers, considering the network resilience to handle intentional attacks. Once again, I'm using a sample network to exemplify the concept of network resilience and attack. So in this sample network, if we remove the node 0, the largest connect component that remains loses one node. So the impact of removing one node, the network loses one node. So impact is null. But if we remove the node 5, we break the network in two parts. So the largest connect component, this one, lose 6 nodes. The one we remove and other 5 that are here. So the removal of the node 5 breaks the network, and more than the large connect component loses more nodes than the one removed. So this node causes a greater impact. We show this resilience to impacts in a plot, and here we have the chromatin organization super pathway. This black dotted line represents no impact. That is, when we remove 10 nodes, the large connect component loses 10 nodes, so no impact. And we see that the green line is the random removal of nodes from the set of node drivers, and the red line is the random removal of genes from the set of drivers. Since we use random removal, we run this 100 times, and the vertical line represents the standard deviation among these 100 removals. We see that there is not a big difference between the removal of drivers and no drivers, and they are both somehow close to the null impact. So the removal from these drives does not break the network a lot. The blue line represents the removal of hubs, that is the node with the highest degree at the time. Okay, so the removal of hub, as expected from the literature, breaks the network a lot faster than the random removal of other nodes. So comparing with the gene expression and the all superpaths, remember that this is the consensus network, we see somehow a similar behavior between these three superpaths. Not significant difference among drivers and no drivers, and a significant impact on the removal of hubs. Now we have another three sets of superpathways: the DNA repair, reproduction, and development biology. And in this case, the three networks show a remarkable resilience to attacks, even to hubs attacks. And the networks barely break, even when we remove drivers, no drivers, or hubs. And lastly, we talk about these two superpathways where we see a differentiation between drivers, no drivers, and hubs. As you can see here, and circadian clock, we have a different behavior. And in the programmed cell death, it's a very important pathway to cancer, it's a hallmark of cancer. The removal of drivers break the network more than the removal of hubs. It's something to be further explored. As a conclusion, we observed that there is a significant difference in some centrality measures, especially between clustering and betweens among drivers and no drivers. In the network resilience, we see three groups of behaviors: chromatin organization, gene expression, and all superpathways does not show a difference between the removal of drivers or no drivers. We see a remarkable resilience in development biology, reproduction, and DNA repair, even with the removal of hubs. And we see a great impact on the removal of drivers in the circadian clock and programmed cell death networks. The results show that the superpathways network have a distinct topology among them, and they also have a particular role for drivers. And group of pathways that share similar results in the centrality measures comparison differ on the resilience of intentional attacks. These findings reinforce the need to diversify the topological analysis we make when analyzing networks. And as for future works, we plan to expand the topological study of these networks and the impact of drivers in their structure. Other topological analyses, such as persistent homology, can unveil hidden drivers' behaviors in pathways, like the case of the NR pair. That's the only pathway we did not see a significant differentiation between drivers and no drivers. And lastly, the patterns found between drivers and no drivers can be used to identify and validate unknown drivers. That's all. I would like once again to thank the organizers, the audience, and my co-workers, George, Cynthia, and Ademus. That's all. Thank you.
Thank you very much, Rodrigo, for this nice presentation. So, as previously, I would like to ask for the audience to write down their questions and the comments while we proceed to the next presentation, which I would like to invite the presenter to share the screen.
Okay, can you see?
Yes, we can.
Great. Good afternoon, everyone. My name is Gabriel Siqueira, and I'm going to present to you our work on heuristics for the cycle packing of adjacency graph for genomes with repeated genes. That's an agenda of the presentation. I'm going to start with a brief introduction of the problem with some motivation of why it's important. Then I'm going to show you the adjacency graph structure and some heuristics we propose to work on a problem in this structure. Next, I'm going to show some experimental results on synthetic and real genomic data, and finish with the conclusions of the presentation. The main use of the problem I'm going to talk about today is on genome rearrangement problems. Genome rearrangement is a mutation that affects a large portion of the genome. The number of rearrangements capable of transforming one genome to another is a way of measuring the evolutionary distance between genomes. Such measure of evolutionary distance can be used in many problems of computational biology, as the construction of phylogenetic trees and the detection of orthologous genes. There are many different types of rearrangement events. We call rearrangement model a set of allow events that we're trying to use to transform one genome to another. Some models, for instance, allow only conservative events. Those are events that don't insert or remove genes. Here are some examples of rearrangement events. The transposition, we're going to pick two sequences of genes and swap their order. The reversal, we're going to pick one sequence of gene and invert the order and orientation of the genes. Those two are events that affect a single chromosome and are conservative. Two examples of non-conservative events are insertion and deletion. We're going to insert or delete genes. Two examples of events that affect multiple chromosomes are translocation and double cut and join. The actual way in which we're going to represent the genomes is going to depend on the characteristics of the genome we are trying to compare and on the events we're going to use. Here we're going to assume linear unichromosomal genomes. With this assumption, if there is no gene with more than one copy, we represent the genomes as permutations. In the general case, if there are multiple copies of genes, we represent them as strings. Using the string representation, every character is going to be represented, every gene is going to be represented as character, and the sign, we're going to attribute a sign to these characters, plus or minus, to represent the orientation of that gene. Using this representation, we have some notations to talk about it. We call the size of the string the number of elements in the string. Si is going to be the character in the if position of the string. Sigma S is the set of distinct characters. This is the alphabet of the string. When we define the alphabet, we ignore the signs of the characters. OCC of alpha S is the occurrence of a given symbol in S, so the number of characters in S with the symbol alpha. OCCS is the maximum occurrence of any symbol in S, so the number of occurrences of the symbol that appears most times in S. Here I have an example. We have a string S of size 9. The alphabet of this string is composed with the numbers from 1 to 6. the maximum occurrence of the string is equal to the occurrence of the symbol for, which is 3. For appears 3 times in the string. When using this representation, one important restriction when you are dealing with conservative events is that the strings must be balanced. Two strings are balanced if they have the same alphabet, and the occurrence of every symbol is the same in both strings. This essentially ensures us that we can transform one string to the other by just changing the order and orientation of the characters. So conservative events can transform one string into the other. The main event I'm going to focus on is the reversal event. This is an event that inverts a sequence of genes under orientation. So here I have a generic application. I have a reversal ij being applied to S. So this reversal is going to invert the sequence that goes from position A to position J. And all the order is going to be inverted, and also the signs. So if a character has sign plus, it's going to receive a sign minus. If it has a sign minus, it's going to receive a sign plus. Here I have a more concrete example. We are applying the reversal for 6, so the sequence from position 4 to position 6, minus 2 plus 4 plus 4, is going to be inverted. It's going to become the sequence minus 4 plus 2. We are going to use this event to define the reversal distance. The reversal distance is the minimum number of reversals necessary to transform one string into a dollar. In this example, we need 3 reversals to transform S into P. So the reversal distance between S and P is 3. Actually, finding this distance can be done in polynomial time with an exact algorithm if we are dealing with permutations, if there is no more gene with more than one copy. In the general case, when there is multiple copies of some genes, we have an NP-hard problem. The best known approximation known for this case is 16 times the maximum occurrence of a given symbol in the string, and an algorithm of O log of size of the string times log star size of the string. Those algorithms have a high approximation ratio, so it is interesting to try to find good heuristics to improve the solution in practice. For instance, an algorithm from the SOR framework that solves the reversal distance problem issues approximations only in some restricted cases, but it uses some heuristics to improve the solutions in the general case. Here we are going to use the adjacency graph structure to propose some heuristics that try to be even better in practice, even if they don't guarantee an approximation factor. This adjacency graph structure wasn't a structure proposed to work on permutations, but it was adapted to work on strings. And in strings, you can use it to find a way of encountering an estimate to the reversal distance between the strings. That's how we define this structure. We're going to have 2 strings, S and P, and we're going to duplicate every character of these strings. So a given character of sign plus is going to become 2 new elements, one of sign minus, one of sign plus. A character of sign minus is also going to become 2 elements, but the first of sign plus, the second of sign minus. We do this for every character, so plus 1 becomes minus 1 plus 1, plus 4 becomes minus 4 plus 4, minus 2 plus 2 minus 2, and so on. The same thing for the characters of P. Next, we add 2 new elements to the beginning and the end of both sequences. We add the element of sign plus, I'm using here plus 0 in the beginning, and we add an element of sign minus in the end. Here I'm using minus 5. Those elements are going to be the vertices of our graph. The edges of the graph are going to be of 2 types. The first type are the black edges. Those edges are going to connect elements that are adjacent in the sequence, but do not come from the same character of the strings. So plus 0, minus 1 are adjacent, and they do not come from the same character, so I have a black edge between them. Minus 1 and plus 1, they are adjacent, but they come both from plus 1, so they won't have an edge connecting them. We'll do this for every adjacent vertices. And next, we add the gray edges. The gray edges are going to connect the vertices from the sequence above with vertices from the sequence below. And they connect it to vertices if they have the same sign and the same symbol. So plus 1 is connected to all plus 1 below, minus 4 to all minus 4 is below, and so on. Those gray edges, they have a special type of relationship. We call 2 gray edges twins if they connected characters, they connect vertices coming from the same character. So, for instance, this minus 1 and this plus 1 both come from this plus 1. And this minus 1, this plus 1 both come from this plus 1. As they are connected by these 2 edges, those edges are going to be twins. Same thing here, this plus 4, this minus 4 both correspond to this minus 4. This plus 4 and minus 4 correspond to this 1. Those 2 edges are going to be twins. We are going to use this structure to define an alternating cycle packing. An alternating cycle packing is a set of disjoint alternating cycles that cover the graph. An alternating cycle is a cycle that alternates between black and gray edges. This alternating cycle pack must respect two restrictions. The first one is that every vertex belongs to exactly one cycle. And the second one is that if we have a gray edge belonging to some cycle, its twin must also belong to some cycle. The only exception are the two gray edges on the extremes, the one connected to zeros and the one connected to fives, that do not have twins. With those two restrictions, we can use a cycle packing to encode a mapping of our strings into permutations, because the twin edges allows us to create a relationship between the characters of the string above and the characters from the string below. So for instance, this plus 4 is connected with twin edges with this plus 4. So we can create a relationship between them and use a different symbol to associate them. Here we replace the 4 with a 4 prime to indicate that as a symbol not yet in the alphabet. We can do the same for the other 4. The other 4 is connected by twin edges with this other minus 4, this minus 4, and this minus 4. I'm replaced with a different symbol, 4 with double prime. If I do this for all characters that have multiple copies, we can now distinguish between the copies of a given character, and we were able to transform our strings into permutations. As I said before, when you have permutations, you can calculate the reversal distance using an exact algorithm in polynomial time. So this reversal distance between the permutations is going to be an upper bound on the reversal distance between the strings, because the sequence that transforms one permutation to the other can also transform one string to the other. The question now is, we have multiple different cycle packings. Each cycle packing is going to give us one reversal distance between the permutations. What we want is the best lower bound, the shortest distance we can find. The question is how to find this. It turns out that the number of cycles is related to the reversal distance. We have to maximize the number of cycles in order to try to minimize the reversal distance. The problem is that maximizing the number of cycles in a cycle packing is also an EPR problem. But we're going to try to use some heuristics to find good packages with a large number of cycles. We developed two heuristics to this particular problem. The first one is one based on random packings. You're going to use a procedure that generates the packings with some random approach, using some grid strategy. This is the BFS packing procedure. I'm going to explain next slide. This is going to construct a set of R cycle packings, a set M, and we're going to pick from the set the cycle with the highest cardinality, the largest number of cycles. The actual procedure is as follows. We are going to pick one vertex from the graph, not yet in the cycle, and apply a breadth first search to find the smallest cycle that have this vertex. We added the cycle to the solution, and we find a new vertex, not yet on the solution. Again, we run a BFS, find the cycle, and we keep doing this until we cover all the vertices. Just have to be careful because we have to keep the twins restriction. So while we are running the BFS, we must be sure that every vertex that is included in some cycle will have its twin eventually in some cycle as well. The next heuristic is going to be a base. We use this one as a basis, but use some more sophisticated ideas. This is a heuristic based on genetic algorithm. It's going to start with an initial population of P individuals. Those are going to be generated by the random packing heuristic. But then we're going to generate new individuals by having multiple new populations. Each new generation that creates a new population is going to have the following steps. We're going to do a selection to select P over 2 pairs of individuals. The selection is done by tournament, so we always pick 2 pairs and compare them, pick the one highest cardinality, select this pair. Then again, pick 2 pairs and keep doing this until we have P over 2 pairs of individuals. For every one of those pairs, we are going to apply a crossover operation to construct 2 new individuals. Apply the operation twice, construct 2 new individuals. With this, we'll have P new individuals in this new population. We're going to apply a mutation to every one of them. This mutation is going to have a chance of changing some of those solutions. Then, just to finish, when we have the final population, we're going to apply a last step based on elitism to ensure that the best solution known so far is kept on the new population. So we compare the worst solution from the new population with the best solution from the previous population. If the best solution is better than the worst solution we know now, we replace this worst solution with the best one. This is just an example to explain the crossover and the mutation operations. We have 2 cycle packings, age and age prime, and I'm going to apply a crossover between them. The crossover consists of picking cycles from one of the two solutions. We're going to pick one of the solutions at random. We're going to pick cycles from then and add into the new solution until we no longer have cycles in the original solutions, or the new solution already have all its vertices covered. So in this example, I'm picking this blue and this green cycle from H, and this yellow and this red cycle for H prime. If there's still some, if you ever try to pick all the cycles, there is still vertex not yet covered, we're going to use the BFS packing procedure to fill the rest of the cycles. In this example, this black cycle comes from the BFS packing procedure. Next, we apply the mutation. The mutation consists of trying to eliminate every one of the cycles. There is a mutation rate that controls the chance of this cycle being eliminated. After some cycles are eliminated, we apply again the BFS packing procedure to insert new cycles. Here, in the mutation, eliminate the black and the yellow cycles, and 2 new black and yellow cycles were inserted. And those are the heuristics. We performed some experiments to see their applications in practice. We created a database of simulated genome with 1,000 pairs of strings. We divided those in 10 sets of 100 pairs each. To create every pair, we produce a source string by randomly selecting characters from an alphabet. Then we shuffle it to produce this target string. The size of the alphabet that we use to produce those strings is actually what distinguishes the different sets. And to assign the signs, we assign the randomly, plus or minus for each one of the characters. We're going to run all heuristics in this data set. We also run the algorithm from the SOR framework to the reversal problem. This algorithm also produces a cycle packing, so we can compare the size of the packing found by our heuristics and by this framework. Here I have some results in this table. I'm showing the average cycle packing returned by heuristics or by the SOR framework, and also showing the execution time of every algorithm. What you can see is that our heuristics, they have a more expensive execution time because they have to produce multiple cycle packings, but we are able to increase the number of cycles in the packings. We see that compared to the heuristics, the best one is genetic algorithms. It was better than the the random packings running in a shorter time. Here I'm showing the random packings with 1,000 packings. Genetic algorithms produce 1,000 packings, and genetic algorithms producing 10,000 packings. If we increase it from 1,000 to 10,000, we clearly increase the execution time, but we also could improve even more the solutions. Just to show that these results reflect on the reversal distance, I have here the same results, but using those the packings found with the heuristics to encounter the reversal distance, using the idea of mapping strings into permutation that I showed before. Using this, we see the same relationships. The genetic algorithm was the best, found the best distances. Remembering that in distances, the smallest distance is better. So JAL is best. JATIN could increase the number of cycles to improve the distance. Those are some results in simulated genomes. We also performed some results on real genomic data coming from the Cyanorac 2.0 system that contains a total of 97 genomes of cyanobacteria. We use our heuristics to calculate the reversal distance between every pair of genomes, but as we are dealing with real genomes, they not necessarily are going to be represented with balanced strings. So we need a preprocessing step to eliminate some of the genes, so the resulting strings are balanced. We tried something simple just to show that the heuristics could solve the problem. Even with a simple preprocessing step, we just kept the first S copies of each gene in the genome, where S is the minimum number of copies in both genomes. So if there is a genome with 10 copies and another with 5 copies, we're going to keep 5 copies in both. We apply heuristics to calculate the reversal distance between every pair and use those distances to calculate, to produce a phylogenetic tree using the circular order reconstruction method. The time to calculate the distance for every pair was 90 seconds per pair on average, so a reasonable time for a real genomic data set. Using the phylogenetic tree, you compare it with the tree presented in the paper that proposed the Cyanorac 2.0 system. We compare those two trees with a method based on maximal agreement tree. That is the maximal agreement tree is the biggest tree that is common in both trees. This method has size 32, which gives us a p-value of 3.47 times 10 to the minus 19. This p-value indicates the probability of the trees being equal by chance. So the lowest the value, the better. So those three are reasonably similar between them. Here is a picture of the tree we produced. You can see the clades well grouped using our distance. Just to conclude the presentation, we propose here two heuristics to the maximum cycle packing problem. This cycle packing can be translated into the reversal distance between genomes. We perform some experiments to show the application of this strategy in practice. We also see that in the simulated genome dataset, our heuristics were significantly better than the surf framework, just given an extra cost on time. As future works, we can test the heuristics to other rearrangement problems. For instance, we can try to apply them to the transposition distance. The idea would be the same using the cycle packing. We also could apply to reversal distance, but now consider unsigned strings, where we ignore the signs of the characters. Again, we could also adapt to include non-conservative events, like insertion and deletion. This would eliminate that pre-processing step necessary for real genomes. But this adaptation needs some changes on the definition of the cycle packing, of the cycle graph. Other thing that could be done is test other heuristics based on other common meta heuristics, such as gradient Ramsey search procedure, tabu search, or simulated annealing. Although this is the presentation, thank you all for the attention. That's what I have to talk about.
Okay, thank you very much, Gabriel. Very nice presentation. So, again, I ask you for the audience that write down their questions. We have some questions there already, but before I read them, I have my own questions, and I'm gonna make them first. So, David, if I understand correctly, you find simultaneously vertex and edges labeled trees to combine orthology and exnology by combining the two strategies, symbolic ultrametrics and Fitch maps, right?
Mm-hmm.
Okay, I confess that the talk before yours from Mark Helmut helped a lot to understand this, and I have a particular interest in this work because I'm starting to work with ADT genes. So I was wondering if you have any plans for a prototype or something like that to make this a real tool.
Yes, definitely. We're not yet completely done with figuring out the details of the math to actually make it a tool that can be applied. But at the moment, as I said in the last slide, we're trying to figure out how we can actually get this direct xenology relation. The first results that we've seen in simulation seem to be promising that we can actually get this from data as we mentioned it in the theory.
I see.
So next step would be real life data.
I hope so. Thank you very much. Well, also for Gabriel, well, I just was wondering that you showed in your presentation that this work is valid for genomes which have only one chromosome, for example. It's just a linear genome. And in your tests, you took genomes from a database from Cyanorak, right?
Yes.
I see. So, and also you said that you make some filters for these genomes, but I was wondering if you when you filtered these genomes to make your data set for testing, have you considered to remove the eventual plasmids from genomes?
Not yet. It's an interesting idea. We didn't consider that, but could be a way to do that also.
Yeah, I see. I just was wondering that because maybe it can affect the results of your tree, because the plasmids are in the genome, but they are not considered as the genome by itself, and maybe you can be testing something over plasmids that are not part of the genomes, and maybe it can affect your results, but not your work, of course, just the results of your uh, the 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 results of your experimental tests, right? So it's a thing I was wondering about. And uh, for last, I would like to ask for to Rodrigo. Uh, well, I think the nature of relationship between the genes can be diverse. Maybe a gene can be related to another one as in a signaling pathway, for example, inhibiting or enhancing some particular biological process, right? And well, you presented uh, difference between topological perspectives when you do something like removing a gene or wherever. And I was wondering also if maybe the biological explanation for those results that you presented in the I can remember the title of the slides when you have the box plot.
The comparison between centroid distribution.
Is exactly. So why in some pathways the you have different results? Maybe they are related to the nature of the relationship between the genes. So have you explored this perspective or not? If not, is it in your plans?
I don't know if I really get the question, but
Okay.
you are saying that there are different kind of edges. Is this because
Yes, yes,
there is different kind of edges?
exactly.
As we said, we extract a list of genes associated to path, just a list. To
Just a list.
transform it on a network, we need a way to say this. There are neighbors. And you use the reactome functional interaction that is one of the many protein-protein interactions that exist. Okay,
Oh, I see. So you are considering protein-protein interaction.
and that
Is
is
this the
from
type of?
reactome. And the reactome functional interaction is made using the interactions the database found on pathways. So it's a protein-protein interaction, which differentiates the reactome functional interaction from others protein interactions is that they are concept thinking about pathways edges. So that's why we use them. Once again, so the network we end up is from is an induced subgraph. But in the reactome functional interaction, there are four types of edges. I don't remember all by name, but we have. Sorry, I can't remember the name of edges right now. But there we can change the topology, looking at, or let's just consider edges that
Particular
indicates
type.
a type of association, and then you have another type of topology.
I
That's a
see.
study that also can be done.
Yeah, I think this is a very interesting strategy because these types have a biological meaning. So maybe you can find an explanation for those difference between
we
the pathways.
consider we consider this, but if I remember, we not put this in the paper. So as when we are exploring the data, okay, and we find that around 18% of the edges are the same type, and the
Mhm.
other three types divide from 13%. So we kind of say, oh, the network from 10 edges, 8 are from the same type. So we just keep our edges as the same.
I see.
And another thing, just to increase the question, the edges also have a score of reliability, let's say. So there are a score on the edge. Besides the edge indicates what kind of relation it has, it also have a score,
I see.
and then we can cut the edges by the type of score. It's once again we lead to another type of network.
So you have a threshold, could have a threshold for cutting some that's not
Yes.
attend to any criteria, right?
For example, there is another database that is string database.
Yeah,
That's
I know.
another a different. They use threshold. They have many more edges, but the score starts at 0.4 and goes to 1, if I well remember. And Hackathon already did this cut at
Mhm.
0.7, if I believe. So they already give a higher threshold for any edge they make available. And we use the default settings for the network. But there are some studies that use this network that increase the, the score.
Perfect. Thank you very much. So let's take some questions from the audience. First of them, it's a question from Marcelo Reis, from Butantan Institute. He asks to Rodrigo, nice talk, congrats. What do you think about the relevance of dynamic events in signaling pathways? Example, post-translational modifications. And if it is the case, how to incorporate them into your topological characterization analysis?
It's a great question. The answer will be similar to the one I did to you because it. But I don't have this information. In your question, I could have made these thresholds, but as far as I know, I don't have the time. Let's say because that the dynamic process, I don't have the time of the edge. But if maybe there is this information on databases, or if anyone has this type of information saying that in a certain moment of the cycle, there are two genes are connected, and then we can. There is a lot of approaches and tools to work with dynamic networks. Okay, we use
Yeah.
this work static network, but there is a lot of things we can do with dynamic networks. And for the simplest thing we can make is, let's say, we define 3 times, and each time we have different edges. So we end with 3 different networks, and we can compare the centrality measures, we can compare the resilience to attack, and we also can analyze how they evolve. So when the delta 0 to delta 2, for example, how they change with the time. But once again, we need the information from this. If we have this information, it would be really interesting to investigate.
I see. And Marcelo Reis, still another question. For very small instance, is it possible to compute an optimal solution, which could then be used reference for assessment of your method's performance?
Oh, yes, definitely. In fact, if the solution is small, the instance is small enough, we could run or at least it could produce all possible cycle packings and would find an exact solution. Probably not the best way to do this in an exact algorithm, but yes, we could use exact solutions to small instance to compare with the heuristic in small cases, and in larger cases, we use the heuristic to find the solution.
Very well. Another question, and this time from Renato Oliveira, from UFMG. So he congrats you, Gabriel, and asks, have you considered to use your solution to analyze the high rate of rearrangements present in mitochondrial genomes of plants? And
I,
okay, go on this one.
okay, I have not, but interesting idea. We definitely want to look into this.
Okay, and the next one, also from Renato Oliveira, is the rearrangement is high, even in mitogenomes from the same species, and your solution could be shed some light into the patterns that plant. Oh, it's not a question actually. Plant mitochondrial genome seems to follow, hopefully. So I think is that. Marcelo Reis also congrats you. he says he forgot to do it before. And if we have no more questions, I think no. So I would like to thank you all very much for your presentations. It was very instructive for me, including. I'm sure for everyone who watch it and will watch it in the future, and because they it will be available on YouTube. So thank you very much, David, Charlie, Gabriel, Siqueira, Rodrigo, Henrique Ramos. Thank you very much, and I end this session here. Thank you all. Today, in a moment, 2 p.m., we're gonna have the rescheduled session, which will be with Dr. Angelica Brian from National Genomic Laboratory for Biodiversity of Sinvestav, Mexico. She will talk about evolutionary genomics of ancient plants and their symbionts from species to metabolites. It's going to be today, 2 p.m. It's the talk that we should have yesterday, but it was rescheduled for today. Again, thank you all, and I finish here this session. Goodbye.